Each one of us started out as a single fertilized egg. Our cells repeatedly divided, so that each of our bodies now consists of about 37 trillion cells of 400 types. How do cells with the same DNA differentiate to form so many different types? In order to understand this, a comprehensive understanding of the relationships between DNA, RNA, and proteins is needed. Scientists used to imagine that if all the information in the genome were to be understood, all genes would also be understood, and the mystery of life would be solved. They therefore desired to read and interpret the human genome, and the Human Genome Project was launched to that end in 1990, starting in the USA. The Japanese research institution that played the main role in human genome analysis is RIKEN. Even if we are able to estimate the number and types of genes that humans have, based on the Human Genome Project, we cannot know the genes that are active in each cell. Dr. Hayashizaki considered that we had to know which regions of DNA are actually transcribed to RNA in an actual organism in order to comprehensively understand the mechanisms of nature life. In 2000, RIKEN launched FANTOM, Functional Annotation of Mouse cDNA, an international research consortium. Researchers from various countries met and worked together to assign functional annotations to the mouse full-length cDNA. More than 20,000 mouse cDNA strands were annotated, and the results were published 2001 in the top scientific journal, Nature. This was so revolutionary that even the groundbreaking Human Genome Project used it as a reference. In FANTOM 2, launched in 2001, new technologies enabled the collecting of very minute amounts of cDNA. These increases in precision of detection have raised unexpected mysteries. The minute amount of RNA previously overlooked was found in large quantity. In this large quantity were many RNA sequences that do not code for proteins. This RNA is termed non-coding RNA.
ノンコーディングを出しているゲノムの領域がですね、ガラクタ DNA と言われたところから全部それが出てきた。驚きというかですね、今までみんなは何やってたんだっていう。
What is the purpose of non-coding RNA? Why is this RNA transcribed from regions of DNA that are not genes? Further study on RNA is needed in order to get to the bottom of this mystery about life. The advent of new technology provided a big clue. This new technology is called cage technology.
The development of cage enabled us to map all of those elements, the gene switches and long-range regulatory switches.
And we know from research that cancers and other diseases are also regulated and controlled by this non-coding RNA.
Non-coding RNAs work together in a network to regulate the behavior of cells.
Non-coding RNA is definitely not just junk. On the contrary, it bears the important role of controlling the flow of genomic information, rather than coding for proteins. If the general functions and actions of non-coding RNA can be elucidated in FANTOM 6, this may lead to a revision of the definition of gene.
ファントムデータがなければ私たちの研究は随分遅れてですね、きっとアメリカやイギリスで、iPS 細胞と同じ細胞が作られて名前も全然違う名前になっていったんじゃないかなと、そんなふうに考えています。今後もですね、ファントムのようなインフラストラクチャー、縁の下の力持ち的な活動が日本でもしっかり支援継続されるように願っています。
Now with genomics we can understand a little bit everything at a higher resolution, so we can have a broad view at higher resolution. With this one we can probably enter the phase of better diagnosis and better therapy.
やっと分かったと思うとですね、新たな疑問が次の世界が開けるんです。生物っていうのは多分ですね、まだまだ我々が知らないことの方がはるかに多い。
DNA, RNA, and proteins work as a complex network. It seems the mysteries of life are hidden there. What is life? RIKEN will continue to try and find answers to the questions that we humans have always held.